friends, today we are going to study about laws of thermodynamics. So first let us study what is thermodynamics. Thermodynamics deals with the energy transformation in the system. It tells us whether a particular physical process is possible or not. It deals with energy and work of the system. What do you mean by energy? It is the capability to do work. Energy can be given by the formula E is equals to F into D. That is, energy is equals to force into displacement, or E is equals to P into delta V. That is, energy is equals to pressure into change in volume. What is the unit of energy? The unit of energy is calorie. 1 calorie is the amount of heat needed to raise the temperature of 1 gram of water by 1 degree centigrade. 1 calorie is equals to 4.184 joule. The SI unit of energy is not calorie, it is joule. Now, what is universe? Universe means system plus surrounding. Now, what do you mean by system and the surrounding? System can be anything on which we are working. For example, suppose if we are working or if we want to study about the energy production or anything related to mitochondria, then in that case, mitochondria will be our system and everything apart from mitochondria will work as a surrounding. That means mitochondria is present inside a cell, correct? We are studying about the energy production or electron transport system, or how the energy is transported in and out of the mitochondria. So in that situation, the system is mitochondria, and apart from mitochondria, everything present around the mitochondria in a cell, including the cell, will constitute a surrounding. Suppose we are studying about the nutrient uptake of a cell. In that situation, cell will be our system and other things around the cell will act as a surrounding. Similarly, suppose we are studying about a complete human body, intake of food or the excretion process or the transfer of heat. So in this situation, as because we are studying the whole human body as a unit, so in that situation the human will work as a system, the human body will work as a system, and rest everything around us will work as a surrounding. If suppose we are studying about how energy or the matter is being transferred from one ecosystem to another ecosystem, in that situation the ecosystem will work as a system, and surrounding that particular ecosystem, whatever will come, act as a surrounding. So universe constitute of system and surrounding. System depends upon the researcher, what the researcher want to continue his or her study. Whatever the target of the study will act as a system. Apart from that, everything will act as a surrounding. So system is the matter present within a defined region of the space. Rest of the matter apart from the system will act as a surrounding. Now, types of system. Based on the exchange of matter and energy, the system can be of following three types. First is the open system. Open system means where matter and energy both can be exchanged. For example, our biological system. Our body is an open system. We exchange matter also, we exchange energy also. Correct? Every cell is an open system because there occur exchange of nutrients as well as the exchange of energy. The next is a closed system where only energy can be exchanged, not the matter. For example, if water is present in any plastic bottle and it is air packed, so in that situation, only the energy can be transferred. Keep a hot water in a tight plastic bottle, after some time, you'll notice the water will cool down because it transferred its heat from itself to the outside surrounding. There occur exchange of energy, but the amount of water will remain the same inside the plastic bottle. That means no exchange of matter. Clear? Closed system means a system where only energy can be exchanged. So if you keep a hot water in a plastic bottle and cap it tightly, in that situation, after some time you will find that water gets cooled down because it exchanged its heat with the surrounding, but the amount of water inside the plastic bottle will remain the same. That means no exchange of matter. And the third is the isolated system where matter and energy both cannot be exchanged. For example, thermos. When you keep any hot tea or coffee inside a thermos, after 1 hour or 2 hours, if you open the thermos, you find that the tea or the coffee remains hot only. And the amount of tea and the coffee is also not changed. That means the matter is not exchanged as well as the energy is also not exchanged. Heat is a form of energy. So because your tea or coffee remain hot, that means the energy is also not exchanged with the surrounding. So these are the three types of system depending upon the exchange of matter and energy. Open system where matter energy both can be exchanged, for example a biological system. Closed system where only energy can be exchanged, for example if we keep a hot water in a plastic bottle and cap it tightly, in that situation what will happen, the amount of water inside the plastic bottle will remain the same. That means no exchange of matter, but the water won't remain hot for longer time. It will cool down. That means it exchanged its heat with the surrounding. That is a closed system. Only energy can be exchanged. Isolated system, that means an example is a thermos. When you keep your hot tea inside a thermos, after 1 hour, 2 hour or 3 hour, if you open it, your tea will remain hot. That means the tea did not exchange its heat with the surrounding. So no exchange of energy, and the amount of tea also remains the same. That means no exchange of matter as well. What is first law of thermodynamics? The first law states that the energy cannot be created nor destroyed. It can only be transformed from one form to another. Or in other words, we can say the total energy of the system and its surrounding is constant. What does it mean? That means you cannot create or destroy the energy. You can only transform that energy from one form to another. For example, plants. If you consider plant a system, so what the plant is doing? What is the basic function of a plant? To perform photosynthesis. What is photosynthesis? Synthesize food in the presence of light. What is light? A source of energy. So what the plant is doing? Converting the light energy into food. Food means chemical energy. Clear? So plants are doing photosynthesis. So photo means light. Synthesis means they are synthesizing food. Light is a form of energy. Food is a form of energy, that is chemical energy. Because food, for example, if we take like glucose, C6H12O6, it's a chemical compound. So what the plants are doing? They're converting light energy into chemical energy. Correct. That means the energy of the universe remained in the universe only. It's just that from the surrounding it entered the system. Sometimes from surrounding it entered inside the system. Sometimes from system it entered into the surrounding, or from one system it transferred to another system. Clear? That means energy is present in the universe itself. It is not leaving the universe. It is only traveling between system and the surrounding. So the total energy of the universe is constant. It is going nowhere. Clear? So energy cannot be created nor destroyed. It can only be transformed from one form to another. Now, what do you mean by state function? State function means any function which is independent of the path followed. That means any function which depends upon the initial state and the final state of the system is known as state function. Say for example, this car. The car has to travel from this green house to the blue house. Green house to blue house, when the car is traveling, if it is not important for us whether the car follows a straight line path or a curved path, when the important for us is the car reach the blue house or not from the green house, these type of functions are known as state function. Clear? When the path followed is not important. What is important? The initial state and the final state of the system, those functions are known as state function. The examples given are pressure, volume, internal energy, enthalpy, entropy, and free energy. These are the functions which depends upon the initial and the final state of the system and not on the path followed. How they reach the final state is not important. The important is the final state and the initial state. Now, what do you mean by internal energy denoted by E or U? Internal energy means the total energy content of a system. If the system is a cell, the total energy of a cell will considered as its internal energy. If we are talking about mitochondria, then the total energy of mitochondria will be its internal energy. So internal energy means energy content in a system. Now, whatever our system is, the energy content of that system will be its internal energy, which is a state function. Internal energy of any system can change only if energy flows in and out of the system in the form of heat or work. If heat flows in, that means internal energy of that system will increase. If heat flows out, the internal energy will decrease. Coming to work, if work done by the system, the internal energy will decrease. Correct? Why? Because suppose when you are doing any work, you are losing your energy, right? So work done by the system means internal energy will decrease. Work done on the system means internal energy will increase. Clear? So the energy of the system can increase or decrease, but the energy of the universe is constant. Clear? So these are the methods by which energy can be increased or energy can decrease in a system, but in a universe the energy will remain the constant. This is an example. Here a cup of a coffee, which is a system, and everything around us is a surrounding. So work done by the system means energy will decrease and will use a negative sign. Work done on the system means energy will increase, denoted by positive sign. Heat gained by the system, energy will increase, positive sign. Heat lost by the system, energy will decrease, denoted by negative sign. Now, formula wise, internal energy can be given by E or U. So delta U means change of internal energy, U2 minus U1, that is equals to plus minus Q plus minus W. Why plus minus? That depends upon, in case of heat flows in or flows out. Depending upon that, the sign will come. W, work done by the system or on the system. If work done by the system, we'll use negative sign. If work done on the system, we'll use positive sign. Clear? So delta U is change in internal energy. Now work done can be given by P delta V, that is pressure into change in volume, or F into D, that is force into displacement. Clear? So in case of W, we can write Q plus F into D. In place of W, that is work done, we can write this or this. So that the formula will become Q plus F into D. Now, what if delta V is 0? That means delta V is constant. So the formula is delta U is equals to Q plus minus W. W is P delta V. So we can write P delta V in place of W. Now what we are saying, if volume is constant, that means delta V is equals to 0. So if volume is constant, this will become 0. In that case, delta U is equals to Q. Clear? Now, the biological system does not work in constant volume state. Biological system work in constant pressure. For example, cell division. When cells divide, when it increase in size, correct? This one, they will increase in size and then they again divide to restore their size. So when the size increased, that means what? Here inside internal volume increased. Clear? So biological system does not work at constant volume because volume increases. It works at constant pressure. Now, when we are talking about internal energy at constant pressure, then the internal energy is known by enthalpy. We use another term for the internal energy that works at constant pressure, and that is given by the term enthalpy. Enthalpy is the heat exchange at constant pressure. Now the formula for enthalpy is delta H is equals to delta U plus P delta V. Clear? When pressure is constant, that means this will become 0. Delta U, from here we can get, it is also Q plus minus P delta V. But this will become 0 because we are working at constant pressure. So delta H is equals to Q. Clear? Here also delta U is equals to Q. Here also delta H is equals to Q. So delta U and delta H is the same thing. Just that when we are saying delta U, that means we are generally saying at constant volume. When we are saying delta H, then we are talking about biological system, that is, that works at constant pressure. And the term is enthalpy. Now, what is entropy? Entropy is the measure of randomness or disorderedness in a system. That means the more the system is disordered, the more the entropy will be. The more the system will be ordered, the less the entropy will be. So entropy measures the randomness or the disorderedness in a system. If the system is ordered, entropy is low. If the system is disordered, the entropy is high. Now entropy is denoted by delta S, which is proportional to Q, that means heat. Q is heat, and it is also proportional to 1 upon T, that is temperature. So overall delta S is proportional to Q upon T. Now what are the factors that affect entropy? The first is molecules. The molecule that can vibrate, twist or rotate have more entropy. That means the molecules that are bonded with single bond will have more entropy. If the molecules are bonded with double bond, it will have less entropy. Why? Because single bond involves s suborbital. S suborbital is spherical and small in shape. They can twist, turn and rotate. So single bond can twist, turn and rotate. Whereas double bond involves pi suborbital. Pi suborbital has restricted movement. So the molecules that are bonded with single bond will have more entropy. The molecules that are bonded with double bond will have less entropy. Coming to individual or combined molecules, the individual molecules will have more entropy compared to the combined molecules. For example, glucose will have more entropy compared to glycogen, which is a polymer of glucose. Why? Because combined molecules are bonded. So because of more number of bonds, the molecules become restricted or its movement became restricted. So glucose will have more entropy, glycogen will have less entropy. Next is concentration. More the concentration, more the entropy, less the concentration, less the entropy. In a class, if there are 2 or 3 students, they will be in an ordered state only. If the class will have 10 students, 50 students, 60 students, 100 students, the entropy will increase. The students will be in their random or disordered nature. Correct? So more the concentration, more will be entropy, less the concentration, less will be the entropy. For example, 1 glucose convert into 6 CO2 plus 6 H2O. So here in the left side, 1 molecule, in the right side total of 12 molecules. So that means this will have less entropy and 12 molecules will have more entropy. So more the concentration, more will be the entropy. The second law of thermodynamics. What the second law states? According to the second law, the total entropy of the system and the surrounding will always increase for an irreversible process. Correct? The entropy of the system can increase or decrease, but the entropy of the universe will always increase for an irreversible process. Clear? All the naturally occurring processes proceed towards equilibrium, which is a state of minimum potential energy. So in the system, entropy can increase or decrease, but the universe will always have tendency of increase in entropy. For example, glucose to glycogen. Here entropy decreased. Amino acid to protein, again decrease in entropy. CO2 plus H2O to C6H12O6, again entropy decreases. Correct? So these we are talking about systems. These are the entropy, change in entropy in the system. Correct? Second law states change in entropy in the universe. So what it is saying, in the universe, the entropy will always increase for irreversible process. But in a system, entropy can increase also, it can decrease also. So the formula will become delta S surrounding is equals to minus delta H, that is enthalpy of the system, divided by temperature. What the first law is saying? According to the first law, which states about energy, that is enthalpy in the biological system, we call the internal energy as enthalpy, delta H. According to the first law, if delta H is positive, that means it is an endothermic reaction. If delta H is negative, then it is exothermic reaction, and this reaction is spontaneous in nature. Clear? According to the second law, delta S, if it is positive, it is an exothermic reaction and it is favorable in nature, or it is spontaneous. Whereas delta S negative is endothermic reaction, which is non-spontaneous. So for a spontaneous reaction, what are the situation? Delta H should be negative and delta S should be positive. Clear? Just now we studied that all the naturally occurring process proceed towards equilibrium, which is a state of minimum potential energy. So delta H negative is spontaneous. Correct? Next, what we studied, the total entropy will always increase. So delta S positive will be spontaneous. Clear? So thermodynamically, delta H negative is favorable or spontaneous in nature, and delta S positive is favorable or spontaneous in nature. Now, what do you mean by Gibbs free energy? It is the energy available to do work. All the energy contained in a system is not available to do work. So the energy available to do work is given by Gibbs free energy. What is the formula? Delta G is equals to delta H minus T delta S. What is delta H? Enthalpy, right? That means total internal energy contained in a system. What is delta S? Entropy. So all the energy which is present in a system is not available to do work because some of the energy will lose due to this entropy or randomness or disorderedness which always increases. Clear? So if we subtract this randomness from the enthalpy, then the energy which is remain will be available to do work. Clear? Hence delta G is equals to delta H minus T delta S. Correct? So what is delta G? It is also the energy content, but it is the energy content which is available to do work. Now what do you mean by standard Gibbs free energy? So standard Gibbs free energy means it is the free energy change at standard condition. Gibbs free energy means this is free energy under physiological condition. Standard Gibbs free energy means free energy change at standard condition. It is not necessary that inside the biological system every time the condition will be standard. What we mean by standard condition? Temperature should be 25 degrees centigrade, pressure 1 atmosphere, concentration 1 molar, pH, if we are talking about chemical reaction, then 0. But because we study in the biological system, we take it as a 7. These are the standard condition. So free energy change in this standard condition is known as standard free energy change. But it is not always necessary that in our biological system, always these conditions will be maintained at all the situation. Clear? So Gibbs free energy and standard Gibbs free energy can differ. Now, the formula is the same only, but in case of standard, we use delta G degree is equals to delta H degree minus T delta S degree. If we are talking about biological system, then we take the pH 7, then we write delta G degree as delta G degree dash. Now, in which condition a reaction will be a forward reaction, or it will be a reverse reaction, or it will be in equilibrium? Here, if we see the graph, we'll find that the initial state has more free energy compared to the final state. Thermodynamically, which is favorable, all the processes will tend to attain an equilibrium, which is a state of low free energy. Always every process or every molecule or every system will tend to attain the lowest free energy state. So when the initial state has more free energy than the final state, that means reaction will go in forward direction. A will convert into B easily because the energy of B is less compared to the energy of A. In this case, delta G is negative because A is getting converted into B. B has less energy than A. So in this case, delta G is negative, reaction will move forward and the reaction will be spontaneous in nature. Initial state has more delta G, final state has less delta G. Next graph, the initial state has less delta G, whereas final state has more delta G. Correct? So definitely the reaction will proceed in backward direction. A will convert into B, where A is the final state. That means final state is getting converted into initial state. This is a reverse reaction where delta G is positive and hence the reaction is non-spontaneous in nature. If the free energy of A and B are equal, in that case delta G will be 0. That means equilibrium. In this situation, delta H is equals to T delta S because delta G became 0. So delta H will be equal to T delta S. Now, delta G of forward and backward reaction will be equal and opposite. If I say you that A to B require 2.5 kilocalorie per mole of energy. Say it plus 2.5 kilocalorie per mole. Then, if I do not tell you what will be the delta G if B converts into A, the answer is minus 2.5 kilocalorie per mole. So, the delta G of forward reaction will be equal and opposite. If A to B takes plus 2.5 kilocalorie per mole of energy, then B to A will take minus 2.5 kilocalorie per mole of energy. Now, we know that by first law, that is energy, and by second law, that is entropy, for spontaneity, we have two situations. So if delta H is negative and delta S is positive, then the reaction is spontaneous in nature or thermodynamically favorable. But what if I say delta H is positive and delta S is also positive? So one situation is not favoring spontaneous reaction, another situation is favoring the reaction. Now the reaction will proceed in which direction? Because in our formula of Gibbs free energy, we have delta G is equals to delta H minus T delta S. So we have delta H also, we have delta S also. If delta H is also favoring the reaction, delta S is also favoring the reaction will definitely be thermodynamically favorable. But what if among these one is favorable and another one is not favorable? Then in which direction the reaction will go? That we can calculate by using this Gibbs free energy formula. That answer is only we can get from this Gibbs free energy, because Gibbs free energy utilizes both enthalpy also, entropy also. So if one is favoring the reaction and other is not among enthalpy and entropy, in that case the reaction will be favorable or not, the reaction will happen or not, the reaction will be spontaneous or not, can be calculated using this Gibbs free energy formula. When delta H is negative and delta S is positive, both are favorable. That means both enthalpy and entropically the reaction will be favorable. So this reaction will be spontaneous at all temperature. Always will get delta G value negative. The example is bond breaking. Breakage of bond is thermodynamically favorable at all the temperature. If delta H is negative and delta S is negative, that means delta H is favoring the reaction but delta S is not favoring the reaction. Then what will happen? In that case, delta G can have either positive value or negative value. How? The formula is delta G is equals to delta H minus T delta S. Correct? Here what we are saying, this is negative, then negative, and this is also negative. Now this negative will become positive. So the value of delta H is in negative, delta S is in positive now. Now what will be the sign for delta G? That depends upon delta H is more or delta S is more. If suppose this is 4 for H, 2 is the value of S, delta G will be minus 2. If we reverse this, 2 for H and 4 for S, the value for delta G will be plus 2. So delta G can have both the values. Either it can go with plus or it can have minus. But only H and S is not responsible because T is also there. That means to become delta G negative, which is favorable. Delta G will be favorable when it is negative. So to get this negative sign, if T is less than delta H upon delta S, in that situation, the reaction will be favorable. If the temperature is less than delta H upon delta S, at that temperature, delta G will gain negativity and hence the reaction will become spontaneous. For example, protein folding, DNA renaturation, aggregation of lipid droplets. Next situation, delta H is positive, delta S is also positive. So in this case, entropically it is favorable, but enthalpically it is not favorable. Now, what will happen in this situation? Again, when one is favorable and one is not favorable, in that situation, we depend upon this temperature. In this case, the reverse will become true. If T is more than delta H upon delta S, at that temperature only delta G will gain negative sign and the reaction will become spontaneous. Clear? For example, DNA denaturation. The last situation, delta H is positive and delta S is negative. In this situation, delta H is positive, that means enthalpically not favorable. Delta S is negative. Here, entropically also it is not favorable. So this reaction will remain unspontaneous at all the temperature. For example, bond formation. Always we have to put extra energy from outside to form a bond. Clear? So Gibbs free energy tells us whether the reaction will be favorable or not if the situation of enthalpically and entropically it differs. If enthalpically and entropically any system or any process is favorable in nature, it will be favorable at all the temperature. If enthalpically and entropically any process is unfavorable, it will remain unfavorable at all the temperature. But if either of one is favorable and the other is unfavorable, in that situation, the temperature will determine that whether the reaction will be spontaneous or not. Clear? Now come to third law of thermodynamics. What the third law states? The entropy of any crystalline perfectly ordered substance must approach zero as the temperature approaches 0 Kelvin. So the law is saying that delta G is equals to delta H minus T delta S if the temperature is 0 Kelvin. When the temperature approaches 0, at that time the entropy will become 0. So that means this will become 0. If this becomes 0 means this whole will become 0. So delta G will become is equal to delta H. Gibbs free energy will be equal to the enthalpy of the system. Now heat transfer, for that we should have two objects having different temperature. Then only heat can transfer. The formula is delta H is equals to Cp delta T. Cp is heat capacity. It is constant, but it varies from material to material. So at thermodynamic equilibrium, heat transfer is zero. That means suppose there are two objects, one is hotter and other one is cooler. So heat will transfer from hotter object to the cooler object till both the temperature will become equal. Correct? Once both the substance will get equal temperature, that means equilibrium attained. After that, there won't be any transfer of heat energy. So heat transfer depends upon two things. One, there should be difference in temperature in both the object. Obviously, one should be hotter and one should be cooler. Then only the temperature or heat can be transferred from hotter to the cold one. And the heat capacity of the substance or the molecule in which heat is transferred. It should have the capacity to accept that heat. Then only heat transfer can happen. Next is Le Chatelier's principle. What the principle is saying? Any deviation from equilibrium stimulates the process that tends to restore the system to the equilibrium. That means if in any system the equilibrium shifts or deviates, then a process will start which will work to restore that equilibrium. That means if any process is going on in which the equilibrium has been deviated or shifted, then again a process will start that will tends to restore or reverse back the process so that again the equilibrium should be attained. All isolated system must therefore inevitably reach equilibrium. Now, this is an example. A plus B giving C plus D. It's a reversible reaction. So, equilibrium constant KEQ will be equal to concentration of C into concentration of D divided by concentration of A into concentration of B. If we are talking about substrate to product, so K equilibrium will be equal to concentration of product upon concentration of substrate if the reaction is proceeding in this direction. Okay, one more point to clear. Equilibrium does not mean equal concentration. Suppose A is getting converted into B. If the concentration of A is 1 molar, concentration of B is also 1 molar, that doesn't mean the equilibrium has reached. Equilibrium does not mean equal concentration. Equilibrium means when there won't be any further change in concentration, that state is known as equilibrium. If after 1 hour, 2 hour or 3 hour, suppose if we look and we find that the concentration of A goes down and the concentration of B increase, that means A again gets converted into B. That means this situation is not in equilibrium. So equilibrium should not be get confused with equal concentration. If both are 1 molar, that doesn't mean equilibrium has reached. Equilibrium means when there will be no further change in concentration. That state is known as equilibrium. So if after attaining equal concentration there occur no change, then that condition will be called as equilibrium. If in this condition after attaining equal concentration also there occurs change or there happen a change, that means that is not an equilibrium. So equilibrium does not mean equal concentration. Equilibrium means a situation or a state when there won't be any further change in concentration. Clear? If after attaining 0.5 and 1.5 we found that there is not happening any change in concentration, that means now they attained equilibrium. If after again 1 hour, if we find that again concentration here decreased, here increased, correct? So that means again their equilibrium has not attained. So equilibrium is a state when there won't be any further change in concentration. It does not mean equal concentration. Clear? The value of EQ is more than 1, the reaction is in forward direction. If the value comes less than 1, that means reaction is in backward direction. If it is 1, that means reaction is in equilibrium. How it is related to delta G degree? Here, when equilibrium is more than 1, we are saying the reaction is in forward direction. According to delta G degree, when the process moves in forward direction, when delta G is negative. According to equilibrium, if it is less than 1, reaction moves in a backward direction. Coming to delta G, we already studied, if it is positive, then it is non-spontaneous and moves in a reverse direction. If it is 1, then delta G will be 0. Now there is one formula or a relation between KEQ and delta G. What is that relation? Delta G is equals to minus 2.303 RT log KEQ. Here R is constant, T is constant, 2.303 is also constant. So by solving this, we should remember these values. If we are talking in terms of kilojoule, the value will come 5.7. If we are talking in terms of kilocalorie, the value will come 1.36. So whenever any numerical is given, you have to solve this, then no need to solve full. You have to remember because this is constant. So for this, if you remember the value, you only have to multiply it with log EQ. Clear? Now from this table, suppose you know KEQ value. You can easily find out this delta G value. If you know this log EQ value, you can easily find out this delta G value. Suppose log EQ value given 10 to the power 6. 10 to the power 6 means log 10 to the power 6 if we do, the value will come 6. How we will calculate delta G degree then? The formula is delta G degree minus 2.303 RT log KEQ. In the question you have given KEQ value, that is 10 to the power 6. So log 10 to the power 6 means here it is 6. Now this is already constant. If in question you asked in terms of kilocalorie per mole or kilojoule per mole. So if you asked in kilocalorie per mole, then you have to put here multiply with 1.36. If in the question it is asked in terms of kilojoule, then this 6 you have to multiply it with 5.7. Here, if you multiply 1.36 into 6, you will get this value. That is 8.17 minus. Clear? So, if KEQ value is given to you, you can easily calculate delta G degree Or if delta G degree value is given to you, you can easily calculate KEQ value. Here, any molecule we can calculate the constant in the form of dissociation constant or association constant. For example, hormone plus receptor giving hormone receptor complex. If we are talking in terms of association, that means HR is the product, hormone receptor. So HR divided by hormone and receptor. If we are talking in terms of dissociation constant, here it will be, this is wrong, here it will be KD. If we are talking about dissociation constant, that means here hormone and receptor individually is the product because it is in reverse direction. So H into R divided by HR. So association constant Ka is equals to 1 upon Kd. Next is Van 't Hoff equation. Van 't Hoff equation is the equation that gives us the relation between entropy, enthalpy and KEQ. So we already have two equations, that is delta G is equals to delta H minus T delta S and delta G is equals to minus RT natural log of KEQ. So here also delta G degree, here also delta G degree. So we can write delta H degree minus T delta S degree is equals to minus RT log of KEQ. Transferring this negative sign from this side to right side, it became RT natural log of EQ is equals to minus of delta H degree plus T delta S degree. So natural log of KEQ will become minus delta H degree plus T delta S degree divided by RT. We can write this in this form separately, RT under enthalpy and RT under T delta S. This equation only we can write in this form as well, that is minus delta H degree divided by R 1 divided by T plus delta S degree divided by R. This we have written to bring this equation in the form of y is equals to m into x plus c. Now this equation can be graphically represented as, so here you have to only remember that Van 't Hoff equation gives the relation between natural log of KEQ and T. That is, natural KEQ is proportional to 1 upon T. If we need to find delta H or enthalpy from this graphical representation, then we have to get the value from slope. If we have the value of the slope, we can easily get the value of enthalpy. If we have the value of y-axis intercept, then we can get the value of entropy. Only this you have to remember from this Van 't Hoff equation. Now, next is Gibbs activation energy. So till now we studied about in which conditions the reaction can be thermodynamically favorable or spontaneous in nature. So what are those conditions? If delta H is negative and delta S is positive, the reaction will be spontaneous in nature or the reaction will be thermodynamically favorable. Now, what do we mean by the term spontaneous or spontaneity? For example, oxidation of glucose is a spontaneous reaction. That means glucose in the presence of oxygen converts into CO2 plus H2O. This reaction is spontaneous or thermodynamically favorable in nature because it has very high negative delta G value. Now, if you want to perform this experiment in your home, this is glucose. That means we all have sugar in our home. So does this mean that if we keep a bowl, open bowl full of sugar in the morning, by night we will see all the sugar get converted into CO2 plus H2 and your vessel is empty, or your vessel do not have any sugar left because all the sugar get converted into CO2 and H2. Does the spontaneity means this? If we keep a open bowl of sugar by morning in our home and by night if we are checking it, do you think that we can find all the sugar vanished from that vessel and get converted into CO2 and H2O? Definitely no. So if it is no, then what the word spontaneous or spontaneity means? So the word spontaneous or spontaneity means the reaction is favorable. That means the reaction will definitely complete, but it has no relation with the rate of reaction. It won't say the reaction will finish in the next second, next minute, it will finish within an hour or two. So spontaneity will only tell you the reaction is favorable in nature or it will happen. But when it will finish, it does not tell anything about it. So to know about when the reaction will finish, we must know about Gibbs activation energy. Lower the activation energy, the faster will be the rate of reaction. Clear? Now delta G are additive for sequential and coupled reaction. That means, for example, if any reaction is unfavorable in nature, then how can we make that reaction favorable? So to make any unfavorable reaction favorable, we have to add one more reaction with it. The reaction that can drive the unfavorable reaction. So these reactions will then become additive in nature. The delta G of the unfavorable reaction and the delta G of the favorable reaction are additive in nature. And these type of reactions are known as coupled reaction. So coupled means the first reaction is unfavorable in nature. To make that reaction favorable, what we are doing, we are coupling one more reaction with that unfavorable reaction. If that second reaction has very high negative delta G value, more than the delta G value of the unfavorable reaction, then it can drive the unfavorable reaction towards its product. Clear? Now here, one reaction is given, A converts into B, but it has delta G value of plus 2 kilocalorie per mole. So that means delta G plus means the reaction is unfavorable or non-spontaneous in nature. Now how can we make this reaction favorable? By coupling. Coupling can be done in two ways. The first way is the product of the first reaction should be the substrate for the second reaction, giving another product C. Now B2C has a delta G value negative. Because of its negative delta G value, now this reaction can drive the first reaction. Because of B to C, A will get converted into B. But for that, this negative should have high value compared to this positive. For example, I am telling you, because it is given here plus 2 kilocalorie per mole, A to B. So B to C, if it has the value of minus 4. Clear? So that means then this reaction will proceed forward. A will convert into B because B to C is favorable in nature. This is an example. Conversion of oxaloacetate to malate has a positive delta G value. So it is unfavorable. Therefore, it is coupled with the second reaction where malate is getting converted into citrate, which has a negative delta G value more than this positive. Here it is 7.1, here it is minus 7.5. So because it has high negative delta G value compared to the first delta G value, hence the second reaction now will drive the first reaction. So conversion of malate to citrate is driving the conversion of oxaloacetate to malate. So this type of reaction is known as coupled reaction, where first reaction is unfavorable and the second reaction is favorable. But second reaction should have a greater delta G value compared to the first reaction. Then the second reaction will drive the first reaction and make it spontaneous. Clear? The second way of coupling a reaction is where A to B is unfavorable in nature. Here one more reaction is happening where in this case B is not the substrate for the second reaction. In the first case, the product of the first reaction is the substrate for the second reaction. But here, the product of the first reaction is not the substrate for the second reaction. Here, some other substrate C is getting converted into D. But this reaction is coupled with the first reaction and it has a very high negative delta G value which can drive this unfavorable reaction towards its product. Clear? The example is conversion of glucose to glucose 6-phosphate. It has a delta G value of plus 13.8 kilojoule per mole. To make it favorable, a second reaction is happening, that is conversion of ATP to ADP, which has a high negative delta G value, minus 30.5 kilojoule per mole. Hence, this reaction drives the first reaction. So these are the two ways by which we can make any unfavorable or non-spontaneous reaction to a spontaneous reaction. Actually, we are not making that reaction spontaneous. What we are doing is we are coupling another reaction with it. And because the second reaction has a very high negative delta G value, it is pulling the first reaction towards its product. Clear? Now, one question from my end for you all. You have to tell me, is photosynthesis a thermodynamically favorable or unfavorable reaction? Okay, do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.